Hello, my name is Patrick Blevins, and today we'll be talking about MHC tetramers. This is for exam 3D. First, we're going to talk about the MHC tetramers history. Although they were developed almost 20 years ago, tetramers are just now really becoming widely available and extremely accessible as tools for the study and characterization of antigen specific T cells. A tetramer is a large molecule made up of four subunits. Tetramers consist of soluble MHC proteins, complex with peptide, and are efficient and easy way to identify, isolate, and study antigen-specific T cells from a variety of biological samples. T cells, or T lymphocytes, are important players in the cell-mediated immune response and are normally characterized by the presence of cell surface proteins such as CD4 or CD8. When exposed to certain stimuli, the T cells differentiate into particular subsets. One of these stimuli is an antigenic peptide that is presented to the T cell mostly by two classes of MHC. All nucleated cells have MHC1 molecules, while MHC2 molecules are usually only found on antigen presenting cells, or APCs. The peptide MHC complexes are recognized by the T cell receptor, or the TCR, and the CD co-receptor, CD4 or CD8. In order for the T cells to become activated, both the TCR and the CD co-receptor must bind to the peptide MHC complex. The specific of binding is conferred through the TCR. TCRs are specific to certain peptide sequences, and therefore each T cell will recognize a unique peptide sequence that is associated with a particular pathogen. In a healthy person with a normal immune system, you could see any pathogen that they have encountered in their lifetime will have activated a T cell with a TCR that is specific to a peptide that is present on that pathogen. These T cell clones then multiply in order to help get rid of that pathogen. One property of the peptide MHC complex that has made studying it so difficult is its low affinity for the TCR. This is necessary, though, in order to allow each TCR molecule to bind multiple MHC peptide complexes, and is the reason for the development of tetramers. When multimerized, the MHC complex TCR interaction becomes more stable as the tetramer can bind multiple TCRs on the cell surface, in turn increasing the TCR binding affinity and decreasing the dissociation rate. MHC tetramer makeup. MHC tetramers are made of four biotinylated MHC complexes, loaded with the peptide of interest 8 to 10 amino acids for the MHC1 class 1s and 14 to 20 amino acids for the MHC class 2s. MHC tetramers have four MHC molecules, but they also have other important components. Biotin, which is used to attach the four MHC molecules to beads coated with a protein called streptavidin. A peptide or antigen, which is a protein from a virus, bacteria, or cancerous cell bound on the MHC molecules. The peptide will bind to the T cells that would only be present if a person has the disease that you are trying to diagnose. A fluorochrome molecule A fluorochrome molecule or a fluorochrome or a molecule that grows in fluorescent light to allow the tetramer to be detected. So at the heart of the tetramer in fluorescently labeled streptavidin molecule, at the heart of a tetramer is a fluorescently labeled streptavidin molecule which can bind four biotin molecules. MHC tetramers are made of four biotinylated MHC complexes. There are multiple ways to make these proteins. The most common is the enzymatic method, the heavy chain, the beta 2 microglobulin invariant light chain, the peptide, and the enzyme BRA, are co-expressed in the Escherichia coli and is mixed together with the peptide. There is a 15 amino acid recognition tag expressed on the C terminus or the MHC alpha chain that allows the BRA enzyme to specifically biotinylate one lysine residue within the recognition tag. The monomers are then purified by size and charge exclusion chromatography and multimerization step happens upon addition of the streptavidin. Since one streptavidin molecule can bind four biotins, the result is a tetramer. It is also possible to have multimers, depending on how many biotins that the backbone of the multimer can bind. If somebody was interested in a specific peptide or wanted to use many different peptides, it is possible to make tetramers using an ultraviolet light photolabile peptide that can be displaced by peptide of interest. The method allows for large-scale screening of any peptide interest. Now that we've talked about how tetramers are made, now let's talk about how they can be used. HLA class 2 tetramers are now widely used in studies of pathogen immunity and vaccine development. In evaluation of anti-tumor responses in allergy monitoring and desensitization studies and in autoimmunity. So I put a picture on here of some different things that they use the class 2 tetramers for: infectious diseases, autoimmune diseases, protein therapeutics, tumor immunology, and allergies. Tetramers have been used by researchers to identify and quantify antigen-specific T cells for patient samples, which is especially useful when determining the effectiveness of a vaccine. Um, Sorry about that. It's also possible to use tetramers to guide epitope mapping, more commonly used with MHC class 2 tetramers. Instead of loading the MHC molecules with a specific peptide, they're loaded with a mixture of peptides from overlapping regions of the antigen of interest. These are then divided into pools of 5 to 10 peptides and used to analyze cells that were stimulated with the antigen of interest. Once there is a positive stain, the individual peptides can then be loaded to identify T cells with a particular antigen specificity and the MHC restriction. These cells can then be expanded in vitro and transferred back into the patient. This has been done for treatment of HIV and for CMV infections. All 9 patients who received transplants of CMV specific T cells showed a reduction of the CMV viremia with no observable side effects. One patient that had a chronic CMV infection that was refractory to treatment when antivirals, but was controlled with 8 days of receiving the CMV specific T cell transplant. It is important to be able to control CMV infection in immunocompressed patients, such as those with active HIV infection or cancer patients. Recently, dextramers were used to identify melanoma-specific CD8 plus T cells from a solid tumor and from peripheral blood. This study established the use of multimers and tetramers in samples other than peripheral blood, and there are more similar studies underway across the world. So those are just a few of the different uses that we have using the MHC tetramers. A lot of it's going into helping autoimmune diseases, cancers, infectious diseases. So there's a good amount of studying and research going on using these. This is the reference I used. I used this website right here. And Dr. Blair, if you want to use this in the future, I'm fine with that. Thank you so much, and I hope you have a great weekend.